Uh,
Sir,
Good morning, sir. Good morning.
Good morning. Good morning. I welcome you to the morning session. Today we have Dr. Nishikant Wase, Senior Scientist, Biomolecular Analysis and Facility Core, University of Virginia School of Medicine. Um, he is with us. He would be conducting two sessions today morning and tomorrow morning. Um, First of all, I'm thankful to Dr. Nishikant Wase, who has agreed to deliver the talk in the refresher course for life science teachers. And though it is odd time for him, but he enthusiastically said, "Yes, sir, I will be discussing certain things like applications of mass spectroscopy in biology with all the fellow teachers." So let me give you a brief introduction of Dr. Nishikant Wase. He did his graduation from JB Science College, Wardha. He did MSc in Botany from Postgraduate Department of Botany, Rashtrasanta Tukadoji Maharaj, Nagpur University. Then he qualified NET examination. Then later on he joined teaching career at Nabira Mahavidyalaya, Katul, and then Dr. Babasaheb Ambedkar College, Diksha Bhumi, Nagpur. Later on he received the overseas fellowship and he went to abroad uh, for pursuing his PhD degree. He has completed his PhD from University of Sheffield. Um, then later on, he did his postdoctoral fellowship from University of Nebraska from 2011 to 2015. Um, he was also research associated at University of Nebraska from 2016 to 2019. Right now, he is working as senior scientist, as I told you, at University of Virginia School of Medicine. His area of interest are metabolomics, proteomics, mass spectroscopy, and plant metabiomics. He has completed two projects, major projects, and has published more than 35 research papers. And today he is going to talk on applications of mass spectroscopy in biology. So I hand over the charge to Dr. Nishikant Wase for his talk. Dr. Nishikant Wase, over to you.
Okay, thank you, sir. Thank you, Dongara, sir. Okay, so today we'll talk about this session is kind of a workshop. So today I'll give you a brief introduction, history about this particular technique, and we'll talk about how to do, if you want to do this kind of experiment, what things we have to do and how we can do this experiment. I'll give you certain examples as well of my own studies. And tomorrow we'll have a hands-on experience with the real data acquired in my own lab, and I'm going to share that data with you, and you will see how we can profile and identify and quantify certain amino acids from a maize plant. Okay, so today we'll talk basically about theory and history of the LCMS. LC means liquid chromatography and mass spectrometry based metabolomics. We'll talk about sample preparation for untargeted metabolomics. We'll talk about best practices for consistency and reproducibility because that's most important. And then I'll talk about two important projects which I have done recently. The COVID study is already published, and there is a second study which is snake venom study, which is under review right now. And tomorrow we'll do a targeted quantification of some small molecules. So if we look at the history of metabolomics, or history of identifying certain molecules, it's a quite old history. People have been doing this thing for centuries. Around 2000 to 1500 BC, there are a lot of Chinese doctors. They start using ants to evaluate the urine patient's urine to determine if they have diabetes or not. Around 1500, there is a German physicist called Ulrich Pinder. He wrote a book called Epiphany Medicorum, and in his book, he described uh, several characteristics, features of urines, based on their color, based on their smell, based on their viscosity, and all other features. And based on those characters, he can identify certain diseases using that wheel, which is called as urine wheel. So if you look at the uh, development and history of metabolomics over the year, so around 2000 BC, we know Chinese started using ants to detect diabetes. Around 1500, the urine wheel was described by Pinder, and around 1905, I think someone's mic is open. Okay, around 1905, the first mass spectrometer was developed. It was developed by Joseph Thomson in Cambridge. So he was a physicist, and he developed that technique and the instrument for which he was awarded Nobel Prize in 1906. Then there is a lot of development happened over the period of time. Around 1922, uh, Hans Dehmelt developed a technique for trapping the ions called as ion trap. Again, he was awarded Nobel Prize for that. Then John B. Fenn, who was working in uh, our neighboring city in Richmond, which is a Virginia Commonwealth University, he was awarded a Nobel Prize in chemistry for developing the electrospray ionization. So ESI means electrospray ionization of biomolecules. So before that, mainly people are using mass spectrometry to determine mass of certain metals and other things. But before John B. Finn, no one has ever done anything with biomolecules. So he was the first person who developed the technique and for which he was awarded Nobel Prize. And then in 1959, there is a Japanese researcher from Shimazu Corporation. He developed a technique called as MALDI. That's a matrix-assisted laser desorption ionization. So what basically we do in that is that we mix a mixture of molecules, or you say a cell lysate, or if you break open an E. coli cell, or if you crush uh, Arabidopsis leaves, so we can take that mixture and we can put that mixture uh, with a matrix on a steel plate, and then we bombard that with a laser, which ionize those ions, and then we can detect those ions to determine what is present in that mixture. And lastly, around 1966, uh, Orbitrap is developed by Alexander Markov. And over the period of time, around 1970s, Linus Pauling defined his first metabolomics paper. In 2006, the first human metabolome project was taken on, and around 41,000 or 42,000 metabolites were described in humans. And by 2020, there are more than 35,000 papers are already published. So if you look at how this technique can be used, so you can see from this picture that this technique can basically use in almost all fields, whether it's a human metabolome, whether it's toxicology, whether it's a personalized medicine, whether it's a microbial metabolome, nutrients, disease prediction, diagnostic development, and so on and so forth. So overall, this is a basic technique which can be used in any field to understand biology. So if we come back to see what exactly is metabolomics, so it is nothing but an high throughput analysis of metabolites or small molecules in a living organism. If you look at the uh, s- central dogma of life, we know there are few molecules which are present in any living organism, which is DNA, RNA, protein, and then small molecules. And based on what you study, there are different fields which is called as either genomics, which deals with the study of DNA, transcriptomics, which deals with the study of RNA and RNA expression, proteomics deal with the study of proteins, and metabolomics deal with the study of small molecules. Both the genomics and transcriptomics are sequence-based techniques. That means uh, you take a DNA, the whole DNA or the mRNA, you convert that mRNA into cDNA, and then you fragment them into small pieces. After that, you provide a radio labeled or fluorescent labeled oligonucleotide and use a polymerase enzyme so that it is basically a PCR based reaction, and each oligonucleotide, when it attached to the template, you can sequence it and you can determine what is the sequence of that particular fragment. Okay, so this is basically what happened in genomics and transcriptomics. But in proteomics and small molecules, uh, it's not a sequence based technique, but it's rather a dissociation based technique where we take the protein, we use an enzyme to digest the protein, to make it into small peptides, and then fragment those peptides into its constituent amino acids, and then we measure the mass of amino acid to determine its sequence and then determine its identity. But if you try to think about these molecules in terms of an analytical chemist or a chemistry person, so DNA is basically polymer of 4 nucleotide. Even mRNA is a polymer of 4 nucleotide. So they are analytically very simple to study. If you look at the protein, on the other hand, it's a polymer of at least 20 amino acids. So it started to get more complex, but still there are only 20 amino acids with different combinations. But when we look at the metabolites, there are like thousands and thousands of metabolites, and they have all of them have different structures, and that's make studying metabolites analytically very challenging. And if you look at the different types of small molecules, you have sugars, we have phenols, we have amino acids, we have alkaloids, organic acids, and then non-ribosomal peptides and lipids. And all of them, every class of molecule has a different structure. And that's what make studying metabolites a very challenging task. So basically, if you look at the typical workflow, we first sit together and design the experiment, that how we want to study the whole thing, how we want to design the experiment so that we can understand the biology and we can get and meaningful information from our experiment. Once we design that, then come the part of sample preparation based on whether we are working with plants, whether we are working with microbes, whether we are working with human or clinical samples and whatnot. So there are slightly different ways of dealing with each type of sample. Once we acquire the sample, then we have to extract them. We have to uh, take out all the uh, cellular content inside, which is bound by a cell membrane or a cell wall outside, and then we can use different methods or different techniques to determine their structure, to determine their quantity. The different instrument which can use are either mass spectrometer or an NMR. Again, in mass spectrometer, there are two different types. One is called as gas chromatogram and mass spectrometer, GCMS or LCMS, that is liquid chromatography. Once we acquire that data, then come the difficult part of processing the data and trying to identify what exactly our sample has. We have to identify them, and from that, then we have to determine level of each molecule within a particular experimental group, and then form an hypothesis and then do an experimental validation. So if we try to do an experimental design, so we have to take into account a lot of different things. How is your study design? How are we collecting the samples and how are we storing it? So say for example, if you are like, I think most of us are botanists. So if we are studying maybe a crop plant and we want to study the grain yield. So if you want to study the grain yield, then we have to study not only the grains, but we also have to study the leaf. We also have to study the root. So harvesting in plants is easy. We just cut it with a scissor or a knife, and then maybe we just put it in a tube, and then we uh, put it into liquid nitrogen to flash freeze it so that the whole metabolism is frozen at that moment. And then we try to store them under in -80 so that their metabolism should not change over a period of time until we finish our analysis. For sample preparation, it's very important to check and calibrate your balance and pipettes. We have to use a standardized extraction protocol. So we don't want to use like one method or one quantity of solvent in one experiment and another quantity of solvent in another experiment. That give you different results. So we have to be very careful about how we do it and how we extract your molecules. Then there is a something called a pool QC sample preparation. So pool QC sample preparation means, say for example, if you have multiple conditions, like you have certain plants which are grown in waterlogged condition, and there are certain plants which are grown in uh, high nitrogen, certain plants which are grown in low nitrogen. Okay, so the pool QC sample means you take a small amount of all the samples and you combine together. That's what call as a pool QC sample. And we also add labeled molecules as an internal standard to all the samples so that we can track our experiment over a period of time and when we do the experiment. For the LCMS analysis part, we have to check the instrument, we have to calibrate the instrument. Before we start the instrument, we have to check its mass calibration, its retention time, and its intensity, and all those things. So the main important thing in any experiment is that the size of the metabolism experiment. So for capturing any biological variation, we have to determine the size of any initial experiment. Say, for example, if you look at these two urine samples, one urine sample looks very normal, and the second sample is coming from a patient which has a maple syrup disease. And you can see the urine is very deep in color, and this kind of phenotype is very drastic. And in this condition, if we have a drastic phenotype, then we can get away with less replicates or less number of samples. Another example here, there are two mouse shown here. One is obese and another is healthy. Again, in these two conditions, we can clearly see visually that uh, the phenotypes are quite different. So in this case, maybe you can get away with at least 3 replicates. But if we have a weaker phenotype, then we need more biological replicate so that we'll have a more statistical power, and uh, we can see that difference statistically. So for example, there are certain situations if we have a cultured cells like uh, microalgae or E. coli or any bacteria. So we need at least 5 to 10 different cultures of wild type and 5 to 10 different cultures of a knockout. If you have animal model, like if you are dealing with mouse, then at least 7 to 12 per group. If you are dealing with plants, so say for example, uh, if you are trying to see grow Vigna radiata or any mung bean or groundnut in 2 or 3 different conditions, then we have to at least 7 to 12 in each group. In a clinical trial, control clinical trial. So control clinical trial means uh, an experiment where most of the parameters are controlled by the investigator. So say for example, if you are trying to study maybe I don't know, autism in small kids. So the investigator will try to see that okay, all the kids should be either from 3 to 5 years, and they should be males or females, and their height should be this much, or their food habit should be similar. So these are the factors which are controlled by the investigator. That's why we call it the controlled clinical trial. And for uncontrolled clinical trials, we need more number of patients or more number of specimen. So that's why we have to have at least 50 to 100. And for epidemiological studies like, say, for example, COVID, where in last 2 years when COVID happened, and then there are few pharmaceutical companies like Moderna or Pfizer or even BioNTech or AstraZeneca. So they did clinical epidemiological study on patients with their drug to see how their drug is working to arrest and stop the COVID-19. And if epidemiological studies are not done on a large scale, even you and me, we could have never taken that drug when it came in the market. So these kind of things are very important when we design the experiments. And so, say for example, there are other factors which affect any study. So, say for example, if it's a non-human subject study like mouse, rat, or a rabbit, so we need to make sure that the strain should be same, the growth condition should be almost same for all those non-human subjects. Their housing where they are kept, they should be well ventilated, and they should be same for all those subjects which we are using for our experiment. Their diet should be same. Their time of feeding to them should be almost same, and the environment and timing. Say for example, if you are taking blood from from those mouse, then you should try to take the blood at the same time, or at least just give it maybe an hour, but you try to finish it within an hour, because if it goes a little bit further, there is a circadian rhythm that affect a lot of things. So to avoid that, we should try to collect all those samples within that time frame. If we are dealing with an experiment which involves humans, then gender affects a lot in any study. We have to make sure that the race uh, is same, or at least there should be an equal distribution of race. At least in India, we don't have that problem. But if you go to a metropolitan or a multicultural country like US, you will have Asians, you have Chinese, you have Indians, you have uh, 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 black, you have Caucasians, and there are so many different races. And if you want to do any study, meaningful study, we have to make sure that all the races are represented. That's why you can use that after the experiment. You can use that drug universally. Again, we have to make sure that ethnicity is same. We have to make sure that the age factor should be considered into selecting those subjects, their exposure, what are their habits, do they smoke, do they drink, do they take drugs, do they take any medication. All these things are important while designing any experiment involving human subjects. Similar to that, if you are doing study on a plant, then we have to uh, think about growth condition. What is the light intensity? What type of soil we are using for growing them? What kind of nutrients we are giving them? What kind of exposure they have in terms of their pesticides or insecticides? What kind of and during our experiments, if you are giving them treatment, so the treatment also matters. The other most important thing nowadays people are considering is soil microbes like the rhizobium. Okay, so these are the uh, factors which affect your study, and before you start the study, you as an investigator, you have to think about all these things and make sure that we have all these things taken into consideration before we actually start the experiment. Then comes the important part of sample collection and storage. So I try to give example in both conditions, when if you are dealing with animals or if you're dealing with plants. So if you are dealing with animals, so there should be consistency in collection and processing of your sample. So say for example, if you are collecting blood, and from blood if you are collecting serum or plasma, so it should be common for the entire experiment. We can't have a situation that half of the samples are serum and half of them are plasma. When we store those samples, there should be consistency in their storage. We should use the same kind of vials, same temperature, and we should minimize a freeze thaw cycle, because the more is the freeze thaw cycle, there is a chance that it will change the metabolism with with every freeze thaw cycle. We also have to consider length of the study and age of the sample. Even if we collect the samples, we can't leave the samples for months and years in our freezer, and then after 3 years we will take out the sample and we expect it to be in good condition, and we can do their experiments. Sometimes we have to add certain enzymes, certain additives to the sample to deactivate certain protease enzymes, so that they will not chew on your proteins, and they will not ruin your sample. Similar to that, in plant metabolomics, the harvesting time is very important, because again, as I told you, the sample should be harvested within a short period of time, because the more time you give it, it will start to change. In plants, when we collect the samples, we either use scissor or a knife to cut that part. But cutting that part means that you are inflicting a wounding effect on it, and that wounding effect also make a lot of changes in the metabolism. So to minimize that, as soon as you remove the leaf or a root or an stem or any part of the plant, immediately put it in a tube and then put that back in liquid nitrogen, so that its metabolism is frozen. Once you collect the samples, you can either freeze dry them or using speed vac or lyophilizer, and after that, you people there is another way to minimize oxidation is to flush your samples with inert gas like helium or argon, so that minimize the oxidation of your sample, and it stop ruining the sample if you want to store it for a longer period of time. As I have already talked you about generating a pool QC sample. So pool QC sample means in this condition we have two conditions. One is a control, second is a treatment condition. And in the control condition we have 5 replicates. In treatment condition we have 5 replicates. So the what pooled QC sample is, you take a small amount from each sample in each group and then you combine it together. That we call the pool QC sample, and we use this sample for conditioning of the mass spectrometer, and then we also use this sample to run at a regular interval to check the stability of the instrument and the chromatography. Another important thing in considering this experiment is that sample randomization. Say for example, if you are collecting some plant leaves, and in your experiment you are dealing with a condition where uh, one plant is grown in full nitrogen and another plant is grown in starve nitrogen starved condition, and then you take all the 10 plants from a fully nitrogen grown condition, and then you process them together, and then you take the nitrogen starved plants and then you process them together. That is completely wrong. You should never do that. What is more recommended is that everything should be randomized. You take one from healthy plant, one from the nitrogen starved plant, and so on and so forth. Because of that, if there is an error coming from the person who is doing this task, that error will be randomized and it will distribute it over all those samples, and that minimize that variation. But say for example, on a Monday morning you are doing some processing some sample. So Monday morning is the first day of the week and you are very excited. But by the time you come to Friday and you start doing it, you are already tired. And maybe that might affect. And if you try to do on that particular day one group of samples and you are already tired, that might affect your outcome later on. So that's why randomization is very important, so that whatever variation you cause will be distributed across all the samples. So once you collect all the samples, the next part is sample preparation. Sample preparation means whatever biological material we have to extract it to make sure that we get all those biomolecules out of that biological material. So it has been done with variety of different solvents and variety of different methods. You can use your good old modern pestle. You can use chloroform methanol and sonication. You can use bead beating and so on and so forth. There are various different methods. This slide is very important because this will inform you if you want to if you want to study particular class of molecules, what kind of buffers or what kind of organic solvent you have to use. Say for example, if you look at the polarity index, water being the highly polar molecule, whereas hexane is the highly non-polar molecule, and then there are different solvents in the middle, which include acetonitrile, methanol, ethanol, chloroform, isopropanol, MTB, and so on and so forth. And each combination of each buffer is efficient in dissolving certain class of molecules. And that class of molecule includes ceramides, amino acids, sugars, oxylipins, acetyl carnitine, monoglycerol, diglycerol, fatty acids, uh, PI, PC, phosphorylcholine, and so on and so forth. So based on what class of molecule you want to study, you have to select that kind of buffer system or the solvent for extraction and for as a buffer. So once you selected all of these and we've extracted all our samples, then come the part where we actually start running these samples on our mass spectrometer. Okay, so the first thing we need to do is that we use a test mix, maybe a mixture of standard, predefined standard, maybe a mixture of amino acids or 5 or 6 different fatty acid methyl esters, or a mixture of 10 or 12 heavy labeled molecules. You use that mixture to make sure that your instrument is in good condition. Because from our own experience, we know that each molecule at what time they elute and what intensity they should be. If it's something less, then we can stop it. Maybe the instrument needs cleaning, we can clean it, and then we can resume our experiment. Then we need multiple QC run to make sure that our instrument is stable. And then after 5 or 10 samples, we run a QC sample in the middle. The reason why we do it, I'll come to that later on. So this is a typical example I'm shown here. So this is a lipidomic study. So we profile a lot of lipids in from a mouse uh, kidney, where the mouse are either on a normal diet or on a high fat diet. And there are two variations. One was a wild type and one was a knockout. So you can see here in this, there is a pink dot, which is a QC sample. So since all the QC sample is a mixture of all these samples and it's run at a regular interval, what normally we should expect is that it should be very clustered together, where they should be clustered very tightly. And that's what we have here. The pink dots here in the middle are tightly clustered, which shows that the experiment has excellent quality control. The second experiment here, which is study of uh, retina of mice. So, over the period of, as we age, we started to get uh, problems in our vision. And it's because our all the rods and cones in our eyes started to degrade. Okay, so this was a study we have done where uh, we found a kind of a discovery here because we found an insulin source of insulin in eye. So this is the study we have done, and right now it's running in review. So you can again see here the green dots are the QC samples, and they are aligned exactly in the middle, and all of them are clustered together. So this shows that there was an excellent quality control for this experiment as well. And this third is a study for COVID samples where uh, I think we have close to 125 patients uh, in this cohort. And again, you can see here uh, the two groups are either healthy or people or patients which are COVID positive. And then this is the QC sample, which is again tightly clustered. So this is what we should look for when we try to do a quality control. So you can see here there are like 7 or 8 different molecules like creatine, guanosine, hypoxanthine, leucine, phenylalanine, thymine, and you can see here these samples had been run around 150 samples had been run here, and you can see their peak area is quite consistent except for one or two where we have little blips up and down, but overall all of them are very consistent across the whole run of 150 samples. So this is their peak area, this is their retention time. So retention time means whenever we on a chromatographic uh, parlay, we say that say for example we put a mixture of complex molecules on a column, and it will start eluting at time 1.2 minutes. So that particular molecule has to elute at 1.2 minute or maybe 1.3 minute over the whole range of all the samples. So this is exactly what this picture is showing, this figure is showing. So these are the basic tools for LCMS based lipidomics. So what we have here is only three things. One is an HPLC or UPLC. This HPLC or UPLC is it's a pump based system which has buffers, and it provides the buffers and the sample, it mixed together and it transport to the column. That's where we separate all those molecules, and these molecules are separated based on their polarity, uh, and as they are separated and eluted out from the highly eluting buffer, they are injected into the mass spec, and the mass spec is the place where we detect all the masses. Again, for separation, there are two kind of chemistries. One is called as Hillic. It's a hydrophobic interaction uh, ionization chromatography, or the RP chromatography, which is called as reverse phase chromatography. Based, based on what kind of molecules you want to detect, you can either choose Hillic chemistry or RP chemistry. Again, to detect certain class of molecules in a better way, we have to manipulate the pH of the solvent. We have to add certain salts or ion pairing reagent, which makes our molecule stick better to the column, and we can detect them easily. So this is a classical example where you can see these same molecules which are run on an HPLC, which has a lower pressure, and that's why to detect all these columns, maybe we have to run uh, these samples for maybe 30 minutes, but on a ultra high pressure UPLC or UHPLC or UPLC, we can do that in minutes because of its high pressure. You can see here that their shape of these peaks are very sharp, and they are eluted very early as well. Then come the part here in the mass spec. So within this mass spec, there are different flavor of methods to detect the mass. Okay, so first is called as data dependent acquisition. So in the data dependent acquisition, what we do is that we measure the mass over charge for all the molecules, and then we select our molecule of interest, and then we fragment that molecule of interest by applying high density energy, and then we record that fragmentation pattern. Okay. There is second type of data acquisition strategy, which is called a data dependent acquisition, or which is called as MSN. So here you can see here we selected a precursor which is shown in red. This is the MS1 spectrum of that precursor, and then we took this precursor and we fragment it. So you can see here the precursor which was higher in intensity, now some part of it is fragmented, and now we have a second fragment here which is blue in color. Okay, we can further take that fragment which is blue in color, which is highest intensity, and we fragment again into multiple fragments. Now we got another higher fragment which can again fragment it here into multiple fragments. So the reason why we do it, because on a mass spec the only thing you will get is mass and intensity. That didn't show you a name of any molecule or any character of that molecule. But by repeated fragmenting that molecule into different fragments sequentially, we can go back and deconstruct its molecular formula, and then we can identify that particular molecule. So that's how it works in mass spectrometry. So in the end, we have acquired all our data, and now we want to see what our experiment has, or what kind of molecule we have. Okay, so the basic processing is that we take the raw MSMS LCMS data, and then there are different ways of analyzing it. So these are the different companies who make mass spec, like Bruker. Compound discovery is made by a company called as Thermo. Okay, Progenesis QI is a software from a company called as Waters Corporation. Agilent Mass Hunter is a software used by a company called as Agilent. Okay, so there are different companies which have their own software, and sometimes their software is very rigid. So the problem with these companies is that you can't use Agilent's data on a Waters in software and vice versa. So because of that, there are a lot of people who have developed open source software such as MZMine, MSDial, or XCMS. So these softwares are good in the sense that they can take data from any instrument, any format, and we can work with that. The only problem is that we have to convert that data into an open format, which is called as either MZ data or MZXML. Once we convert that, then we can pre-process the data, and we can come up with our results. So in the end, when we do the pre-processing, the only thing we get is mass, retention time, and its intensity. And from that, we do statistical analysis. We identify what features are important and what features are significantly changed, and then we try to identify those features using different methods. So this is a typical, again, data processing workflow. I shown it earlier. This is another way of showing it. So if we convert our data into an open format, so the first thing we need to do is that do a peak detection. So say, for example, if you can see here on panel number 2, there is a horizontal line. So based on our experience, we determine that what is the signal to noise ratio, and then we put a cutoff on that. So that anything below that signal to noise ratio will be discarded, and we will only take any signal which is above a certain threshold. So that is shown with a green check mark. Once we detect the peaks, the next part is to build the chromatogram. So by using some algorithms, we build this chromatogram into nice Gaussian peak shape. Once we build the chromatogram, then we have to define their boundaries, which is called a deconvolution. So you can see here, we have defined a boundary for each chromatogram so that the software know this is a particular peak, and then we have to look for this particular peak in all our samples. Once we detect the deconvolution part is done, then we do the isotopic grouping, and then we try to align those features from all the samples together. Okay. Once that is done, in some cases, there may be a situation that a particular feature is identified in 10 out of 15 samples. So there are 5 samples where we didn't find it. So what the software do is, or what we try to do is that maybe try to adjust the retention time, or make the retention time window a little bit bigger, and try to find that particular feature. If we find it, then we'll take that value. If we don't find it, then we'll leave it as blank. Okay. And finally, we still have to go back on every feature and do a manual validation. And finally, we can export that data in Excel. So how we do the metabolite identification and annotation? So say, for example, we have this leaf. We extracted that leaf in methanol, and then we run it on a mass spec. And then we detect the full scan mode, where you can see here there are 3 different peaks. One is a very big, and then they are 2 small. So this is called an isotopic pattern. In a naturally all molecules have their isotopic pattern. Say, for example, carbon. So we have carbon 12 and carbon 13. So in nature, carbon 12 is most abundant, which is like 98%, whereas carbon 13 is only 0.2 to 0.3%. So this is what this is showing, the isotopic pattern. And from that isotopic pattern, we can determine its molecular formula. But the problem is that just with the isotopic pattern and molecular formula, we'll get a mass, and that mass can give you maybe more than 1,000 different molecules, which is not good because that confuses us. So the next strategy is to acquire the MS1 data, and also take the MS precursor and then fragment it into different fragments. Now what we have is a feature, and we also have a lot of fragments, and then we try to match those fragments or experimental fragment with a data or a fragment generated from an authenticated standard. If it match, then we say that okay, this particular molecule is 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 what it is in the standard. But to run standard, or provide an authenticated standard is very expensive business. It takes a lot of money, and not everyone have it. But what people are doing over the last 30, 40 odd years is that whenever they acquire data on a standard, they they submit that chromatogram on a public repository like NCBI, or other places. And doing that, we have people have developed something called as experimental library of molecule. Now we can take this library, because now we know that this library is coming from spectra which is acquired from a standard, and we can use this library to interrogate our data and to identify our molecules. If you look at all these are all the databases which are basically used for identification of molecules, such as mass bank, mg cloud. Lipid bank is basically used for identification of lipids. Then there is a cake database which is used for all the biological molecules. This is HMP, which is human metabolome database, biopsych, mona, and so on and so forth. So in a typical experiment, this is what is happened. So you start with an experiment, we start with an experiment, and we get something around 31, 32,000 features. Out of that, we see that maybe 8,000 on 8,000 precursor, we have acquired MSMS on it. After doing a lot of filtering, we left with only 6,000 features, and then we try to match with the reference database, and in the end we come up with only 300 to 400 molecules which are identified confidently. And out of that 350 or 400 molecules, only 10 or 10 to 50 molecules are significantly changed, which can explain the biology. So that's the typical outcome you can expect from any metabolomics or any uh, experiment of this kind. So this is the study which is published recently in 2022, where I was involved. back in 2020, when the COVID started, we had a lot of patients in the hospital in Virginia, and people came said, okay, we want to do this experiment. I said, okay, fine. So what we had here is that there are 52 patients or healthy individuals uh, in the hospital. We took blood sample from them. There are 48 people who had mild COVID. So who were by using qPCR, they detected that they had COVID, but they were mild. So we collected their blood, and there are 25 patients who were on ventilator. So we collected blood from them, and we want to, and the goal of this experiment was to try to compare uh, the metabolome of all these three category of people, and to see what we can learn from them, and can you predict why certain patients who had COVID remain mild, whereas certain patients went to become very severe and have to put on ventilator. Okay, so we collected their plasma, we did their metabolite extraction, and we run it on an LCMS. We use a software called as Compound Discoverer to identify and process the data. And in the end, we did a lot of statistical testing, univariate analysis, multivariate analysis, and metabolic modeling. So you can see here, this is the quality control samples of the 125 patients. You can see here the peak area over a period of 125 sample run is very consistent across all these molecules we have put into the sample. Normally, why it says negative and positive? Because there are certain class of molecules which are detected better in positive ionization mode, and there are certain class of molecules which are detected better in negative ionization mode. So say for example, all the amino acids, nucleic acid components like adenine, guanine, GTP, CTP, cytidine diphosphate, they are detected better in positive ionization mode. But if you look at lactate, lactic acid, fumaric acid, succinic acid, citric acid, isocitric acid, they are all detected better in negative ionization mode. That's why we have to run our samples in both positive and negative ionization mode. And even in both the condition, you can see here that their peak area uh, of the QC samples is very consistent, and the retention time is very consistent, which shows that the quality of the experiment was uh, quite good. So the first thing we do in this kind of experiment is that we take all the data and then we normalize it. The reason for normalization is that we try to reduce the heterogeneity or the variability among the samples, so that uh, all these samples will be homogenized nicely, and then we try to look for features or molecules which are different among these different conditions. Okay, and the first thing after to do is that to try to look to make an overview of that whole data, which is called as principal component analysis. So within this data, we have data coming from 125 patients, and we have at least 500 to 600 molecules. It's a very big Excel sheet, and we don't know how our data looks like. So the first thing we do is that we run something called as PCA, principal component analysis, and it's a dimension reduction technique where we try to, without giving any other information, we try to see whether biologically these groups are different or not. As you can see here, uh, they are not very much different, except for the fact that the patients on ventilator are slightly differently defined, whereas the patients which are healthy and which are mild COVID are overlapping over each other, which makes sense because the patient which are on ventilator has a severe crisis in their metabolism, whereas patients which are mild and healthy, they are kind of there is not much difference. But to tease out their differences, then we do uh, another technique called as partially square discriminant analysis. It's again a, a, a linear modeling technique where we use where we try to identify how these two groups are different, whether they are different or not, number one, and if they are different, then what are the components which are making them different. Okay, so you can see here uh, on the PLSDA among the healthy and the mild patients, even within the healthy and mild patients, we can tease apart those two groups together nicely, and we can identify certain features or molecules which are responsible for this group separation. But when we try to compare these healthy and patients on ventilator, we get a very clear separation that yes, there is a huge difference in the metabolism between patients which are healthy and patients which are on ventilator. In the end, after the modeling, what we identified is that there is a this this pathway of tryptophan metabolism, melatonin synthesis, is highly affected in the patients which are mild which has mild symptoms. Okay, whereas patients which are severe symptoms and which need ventilator, their metabolism was highly affected. The histamine metabolism was highly affected. And just to show some of the molecules which you can see here, that serotonin, there is a huge drop in the level of serotonin in patients which are on COVID ventilator. Again, melatonin, again there is a huge drop of these melatonin levels in the patients which are on ventilators. High hydroxy L-tryptophan, again huge drop in that ventilator patients. L-tryptophan, anthranilic acid, and all those molecules. So these are the biomarkers we identified after our modeling, that these are the typical biomarker where if if you profile any mild patient, and if these molecules are highly reduced in that uh, mild patient, there is a chance maybe in future is that he might go on a ventilator. So that's what this whole thing is about. So in summary, we found that tryptophan metabolism in mild COVID-19 patient paint a metabolic picture of innate immune system inflammatory response. And we also found that there is increased activation of histidine and ketone-related metabolism in severe COVID-19 patient, which show that there is a musculoskeletal degeneration. And we found that histidine plays a key role in activation of serine protease. This particular protease enzyme, which helps the virus entry into the host cell, because the serine protease generally degrades the surface protein, and when surface proteins are degraded, it's make the virus to very easy to enter into the host cell, and that's how the COVID is spread throughout the body and then on the next patient. And there was also increase in the ketone body biosynthesis, which indicate that there is an increase in the fatty acid oxidation, which results in a ketone body formation by the liver. So that was one of the study, and the second study is a longitudinal lipidometabolomic study of envenom mice. So this study we have done in collaboration with a researcher from Brazil. So as we know, a lot of countries like Brazil has a huge forest, and just like back in India, we have a problem of snake bite everywhere. So I think this Bothrops is called a manyar, isn't it? I don't remember the exact Marathi name. So basically, what we did here is that so far, lot of studies has been done. People have known, and people try to study what happened clinically. So whenever we have a snake bite, there will be a degradation of the local degradation of the site where the snake bite is happened, and then there will be some systematic changes. If the snake bite is not treated quickly, the patient might die. But so far, there was no evidence that people have looked into what happened when the snake bites and what happened into the metabolism. So we try to address that problem with this study. So what we did here is that we collected venom from these two snakes, and we injected that venom into this mice. Uh, it was not an intravenous injection, but it was an intramuscular injection because when a snake bites, most of the time it just on the outer muscle. So to mimic that, we have to inject the venom into this uh, intramuscular injection on this mice, and then we collected its blood at 0 hour, 1 hour, 3 hour, 6 hour, and 24 hours. Uh, we collected the plasma, and then we extracted metabolites and lipids from it. But today I'll just talk about the lipids here, and then we run it on the mass spec, did this analysis on it. So again, when we look at the PCA plot of that, so basically here we injected the mice with that venom. We collected the blood. We didn't have any idea what to expect. So this is a PCA plot of the snake called Dabuya. And you can see here, there are these two groups, which is 0 hour and 24 hour, they are clustered together. Whereas all the other time points like 1 hour, 3 hour, and 6 hours, they are quite separate from those two groups. The same thing happened again in Bothrops as well. Again, 1 hour and 24 are clustered together, and the other three are separate from each other. So just by looking at this picture, what we can deduce is that maybe the metabolism, there is a huge effect on the metabolism at 1, 3, and 6 hours, and by 24 hours, in at least in this situation, uh, the mouse somehow managed to overcome the poison effect, and its metabolism come back to normal. On hindsight, it could be a mistake on our part that we use a concentration of 20 mg per body mass weight. Maybe we might have estimated, or we might have injected the venom at a lower concentration, because when a snake bite, maybe the amount of venom goes into that host body is perhaps higher. That's why it dies. So totally, we identified total 120 lipid species in this experiment. And you can see here, the majority of the lipid species are represented by something called as triglycerides. So these are the neutral lipids, which are uh, in most abundantly present in this organism or in this mice. Then we have phosphatidylcholine, sphingomyelin, for LPC, PE, triglycerides, cholesterol ester, and so on and so forth. So this is typical what we can see here. That what we see here is that after the snake bite, the red is represented by Deboya and blue is from Bothrops. So the acylcarnitines are highly increased in the mouse around 6 hours, whereas the ceramides are increased uh, at the first hour and the 6th hour, whereas there is no effect on the uh, from the venom of the other snake Deboya. The major thing which we found is that all the LPE and PC and PE, these are the components of your membrane. All because all our membrane is composed of lipids. So all those PC and PE are the components of the membrane, and this indicate that there was a huge uh, degradation of the membrane, and that's why these lipids are circulating and reduced from the bloodstream. Along with that, we also found that there is an increased triglyceride. That means that there is an increased accumulation of neutral lipids in the bloodstream. So in the end, we try to see what pathways are changed based on pairwise comparison. But this slide is quite busy and I don't think it's readable. So in summary, what we found is that snake venom elicits a strong inflammatory reaction, and it releases damage induced particles from affected tissue. And that's what we detected in the analysis. We observed that both venoms induce an increment in the level of triglyceride up to 6 hours, and then it returned back to its baseline value at 24 hours. We saw that there is a drastic drop in the pool of PC and PE, like phosphatidylcholine and phosphatidylethanolamine, but eventually they again come back to the baseline level. So that's all I have for today. Thank you, and I'm happy to take any questions.
Thank you, Dr. Nishi. It was a
Mm,
wonderful talk. Even a layman
okay,
like me can understand each and every aspect of your talk.
okay. I mean,
You have used a very simple and lucid language to explain each and every aspect of your topic. Um, what you are contributing
thank
to
you,
the science is
thank
a
you,
very
sir.
good thing.
Yeah, yeah, yeah. It's a very tall learning curve for me coming from botany to all these things. You can imagine.
Yes, I do agree.
From botany to this, it's a tall learning curve.
Yeah. Are there any
Yes, sir.
questions from the audience?
Yeah, if you have any questions, please feel free to ask.
Excuse me, sir.
Hmm.
Sir, what method should we follow to study metabolomics using cell lines? Because to stop metabolism in cell line, we have to take these cells from 37 degrees Celsius to minus 86, and it is not an easy step to preserve cells from 37 to minus 86.
No, no,
So,
no, no.
okay.
The reason why we keep it at minus 80 is that so that we because at a physiological temperature at 37 degrees or 40 degrees, all those protease enzymes are already active. Okay, and when the protease enzymes are active, they started to eat all or degrade all your proteins, and that affect your metabolism. By putting that cells first flash freezing in liquid nitrogen and then putting in minus 80 means that you are stopping all the metabolism so that you can preserve that state of metabolism or snapshot of metabolism so that you can study it later. But say for example, if you want to study like do this kind of experiment, I think even Nagpur University has now a GCMS, so you can use that
Yes,
GCMS to study this. I think Badare sir is in charge of that facility, so maybe just talk to him.
yes, sir. Thank you, sir.
Thank you for a very informative and nice presentation, sir. And
Okay.
as we all are very much new to this metabolism, what
Yeah, so
new
I,
topic,
I think for yeah,
metabolism.
yeah. So what we will do tomorrow is that I
Yeah,
have provided. I think Dongara sir might have already given you the link to download the software. So download that
yes, yes,
Skyline software,
to download, sir.
install it, and there are some raw data files. So download, take those data files as well, unzip those, and keep it with you. So tomorrow,
Mhm.
what we will do is that although you haven't acquired that data, but tomorrow I'm going to
Yes, sir.
show you how you can analyze the data and how you can identify a certain class of molecules, and then we can talk about that. It's a kind of a hands-on workshop, so
Yes, sir.
we will learn that thing together.
Okay, sir. Thank you very much. Sir,
Okay.
can we use GCMS instead of LCMS for
Yeah, yeah,
the
yeah, you can use. So
metabolism?
basically, to study this, not only GCMS, you can use
Okay.
GCMS, you can use LCMS, you can use NMR, you can also use TLC plates, you can use paper chromatography strips. That's
Yes.
so TLC plates and paper chromatography plates are the like initial form of chromatography which we have studied in our BSc or MSc
Yes,
classes.
right.
Okay, so
Uh,
basically what we are doing is that we are we are taking a mixture of molecules, putting on a TLC plate or a paper
chromatograph.
chromatographic
Right.
plate as a spot, and then under the influence of certain solvents, we are trying to separate them based on their in the TLC or paper chromatography we use RF value.
Yeah.
Okay, so
Yes.
RF value is kind of equal to what we say in HPLC with the retention time.
Right.
Okay, so the concept is almost same. The only thing is that here the instrument is little bit fancy. That's the only thing.
Right, sir. I have one more question.
Mhm.
During study of corona
Mm.
metabolomics, you have mentioned the certain hormones or certain essential chemicals get dropped down, like you mentioned the melatonin. So,
Yeah,
decreased amount of melatonin, was that related matlab, to drop down in the sleep?
yeah. I think
The patient was
because the
having the
problem.
common problem.
Yeah, I think that the other thing is that because that patient was in a stress condition, and
Right,
if you are in stress condition, if you can't sleep, definitely the melatonin level
right.
will go down. So this is one of the feature of that patient where you have a less melatonin level as compared to healthy patient. But along with that, there are other molecules which are part of that innate immunity cascade, and
Mhm.
melatonin is also a part of innate immunity cascade. And
Right.
because in this situation uh, you have a problem with your immunity, and your immunity is compromised, that's why this patient has to go on ventilator, not the other
Right.
patients which are come out as a mild patients.
Yes. Thank you, sir.
Uh,
Thank you very much.
oh,
Mm.
sir, I have a question.
Mhm.
Uh, Not actually a question. I just want your explanation.
No,
Please,
no,
can you
no.
please
Feel free to ask anything. Don't worry about that.
can you please elaborate on the level of histidine and the serine protease and the COVID infection just before you talk about? Can you please elaborate on that?
No, it was not histidine protease. It was serine protease. So
Serine
see,
protease and histidine
yeah,
level something you.
yeah. So
Uh,
what happened is that histidine plays a key role in activation of serine protease. So serine protease is a protease enzyme which degrades lot of surface proteins. Okay,
Right.
and that facilitate the entry of virus into the host cell. Okay, so and 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 because of that, because we know that histidine pathway was degradation pathway was activated, which means that serine protease was activated, and that specifically in these patients caused this rapid spread of uh, COVID virus into their body. And along with that, we also found that there is increased ketone bodies biosynthesis in those patients. Could be because of the increased beta oxidation, and because of the increased beta oxidation, uh, the ketone bodies will be their biosynthesis will be increased in the by the liver. This
okay.
is a very Vivek. This is a very complex chemistry and interplay of different pathways. We are
Okay.
just scratching the surface.
Sir,
I mean,
another
we
thing.
are lucky. We are lucky that we get this vaccine, but we still it will still take maybe many more years to completely understand how this virus take over your own defense mechanism and play around with it.
Sir, in the same context, I may be wrong, but
Mhm.
as we know that for a COVID virus, it needs some kind of receptor proteins on the cellular membrane. If a serine
Yeah.
protein is degrading the proteins present on the membrane,
Mm.
then how these viruses get recognized on that level, and particularly in the respiratory tract?
Yeah, that's a good question. I don't know. I haven't thought about that. I haven't thought about that. Maybe I can get back to you that later on.
Okay, thanks, sir. Okay.
Hello, sir.
Hmm.
Just a small query. Like of late, we have been hearing of heart attack deaths in the youngsters post-COVID vaccination or post-COVID. So, is it actually correlated or it's just a coincidence?
Mm, I can't say anything about that. But there is one thing I have noticed, specifically in lot of Indian population. I don't know whether it's right, whether it's wrong, but what I've been hearing from a lot of people is that there are a lot of young people who were seemingly healthy earlier. They accident, these days they have started to have problems with their kidneys, started to have problems with their liver, so much so that they have to go for liver transplant or kidney transplant. I'm not saying it's COVID, but there is another aspect to it, that a lot of people have taken these kadas. So, the thing is that a lot of pharmaceutical companies, they spend billions of dollars to develop a drug. Before they develop a drug and before they release into market, a lot of testing has been done on it based on patients and experimental data. What we are doing in that context is that someone told you that, okay, take these dalchini, take these few masalas, uh, put it in a kadai, boil it for half an hour, and then take 2 spoonful every day as an immune booster. We have to understand that what these uh, very constant decoction is doing to our body, because our body can take this kind of abuse at certain limit. After that it will just give up, and that's why people are having problems with kidney failure and liver failure.
But
So
sir,
there are
this
so many things, yes, there are so many things happening. Um, but the thing is that there are things which are not in our control.
but this may not be a generalization. Like not all of them would have taken all these decoctions, etc.
No, but at least I know at least 5 to 6 people who were like seemingly healthy. They don't have any bad habits. No one drinks. No one consumes tobacco, and they have these problems.
Right.
And when I try to ask them that, then they say that yes, I have taken some kara or something like that. So these are the problems people are facing. Then there are people who are taking, like bodybuilders who take protein shakes or the protein supplements, or people who take lot of herbal life, ayurvedic medicine. I'm not against ayurvedic medicine, but lot of in lot of situations these medicines are not actually tested. No one has ever done any clinical trial on it. We really don't know. So these are the lot of different things which affects our body as well. And say for example, if someone is taking this kind of medication, and by taking this medication consistently over 5 to 6 months or or a year, you have already like reduced the capacity of your immune system, and that's why any virus or anything can attack your body very easily.
Or else, just it came to my mind, virus being a protein particle, when it is coming out of
Mhm.
the body, it may be mutated in some or the other form, and these particles may affect the human system in some or the other way. Just a
Yeah, yeah,
thought
yeah,
came to
yeah.
my mind.
It's completely possible, completely possible. The
Like
thing is that
so, uh,
this particular virus was quite new to us, because it's RNA virus, and RNA virus are very immune to rapid mutation. So it can change very rapidly. That's why we have Omicron variant and beta variant and alpha variant. These are nothing but maybe change in one or two amino acids, and it change its form.
right. So when can we get free from this?
These are ongoing studies. People are studying it. It takes time. It takes really time. Normally, if you go back to the epidemiological history, like the Kala Azar or the black fever and other diseases, it took 20, 30 years of work. Then we come up with a drug or medicine or a vaccine. At least we are lucky that after this pandemic started, and within 2 years, we all of us have at least a vaccine.
Right.
In normal course, it could have taken 10 years.
Yeah. Thank you, sir. Thank you so much.
Problem.
So if there are no questions, thank you very much, Dr. Nishikant. I, on behalf of
Thank you, sir.
Rashtrasanta Tukdoji Maharaj Nagpur University and UGC HRDC Center Nagpur, thank you. I'm thankful to you for delivering such a nice and informative lecture um, on metabolomics. And I know that it is very odd time for you. It is morning 2 o'clock there, and
Yeah.
it was your generosity that and your
No,
humbleness
no,
that
no, sir.
yes,
It's nothing like
yes,
that. No, don't mention it. Yeah.
yeah, but you were keen to discuss with all the teachers regarding your
Yeah.
work. So once again, thank you very much, and we
Yeah.
all are looking forward for tomorrow's.
Yeah. Tomorrow's session. Plus,
Yes.
please, all of you, please make sure that you download the software and the raw files, and then we'll work on that. Okay.
एक्सक्यूज में सर हेलो
Uh-huh.
हाँ वो सॉफ्टवेयर जो लिंक भेजी थी ग्रुप पे उसके ऊपर क्लिक करने के बाद सीधा गूगल ड्राइव ही ओपन होता है तो
हाँ,
वो
तो
प्रोसेस अगर हमें
उसमें एक exe फाइल होगी स्काईलाइन करके, तो उसको
हाँ
डबल क्लिक करने से वो ओपन होगा, एंड देन बट यू हैव टू हैव एन एक्टिव इंटरनेट कनेक्शन बिकॉज इट विल कनेक्ट टू दैट सर्वर एंड देन इट विल डाउनलोड few things and it will install it. Okay,
ओके
so
ठीक है
make sure that you should have an active internet connection when you are installing that software. After that, you don't need internet connection.
सर
ओके
उसमें
Hello.
सर रॉ फाइल उसमें से वो ये नहीं हो रही है, इंस्टॉल नहीं हो रही, रॉ
Just a
फाइल
minute.
इज नॉट इंस्टॉलिंग, व्हाट्स द रीजन?
Raw file.
रॉ फाइल मतलब वो रीड नहीं होती है और इंस्टॉल नहीं हो रही है।
नहीं, तो रॉ फाइल तुमको वो रीड भी नहीं होगी, वो मैं बताऊंगा ना कैसे रीड होगी। तो
जी 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 ठीक है सर।
हेलो सर अगर
ओके
आप
ओके
एक बार दिखा दोगे कि गूगल में उस लिंक के ऊपर क्लिक करने
हाँ,
के बाद कौन सी फाइल
एक
के ऊपर
सेकंड मैं
क्लिक कर
दिखाता हूँ आपको।
ओके
Just give me a minute. Huh? Okay, can you see my screen now?
Yes, yes, yes.
Yes, sir.
Okay, so on the screen, can you see that web page I am clicking?
Yes, sir.
No.
यस सर
Yes.
Okay, so go to
Yes,
the data software folder.
So can I see?
ओके तो जो सॉफ्टवेयर नागपुर में डाटा सॉफ्टवेयर फोल्डर है उसके अंदर स्काईलाइन सेटअप exe फाइल है okay so just double click that but make sure that you have internet connection on it will automatically install it just click next and it will install it okay
सर यहाँ पे क्लिक करने के बाद राइट साइड में हु हैज एक्सेस आ रहा है, एक कॉपी लिंक वाला सिंबल आ रहा है और एक लिस्ट के टाइप का सिंबल आ रहा है। वो स्काईलाइन वाला जो आपके इसमें दिख रहा था वो नहीं शो हो रहा है।
ideally everyone should have that access okay मैं एक काम करता हूँ ये सारा को जो सॉफ्टवेयर है ना I'll put it in Dropbox and share that link with डोंगरवर सर और नहीं तो यहीं पे मैं शेयर करता हूँ एक सेकंड मुझे अभी मेरे लैब के कंप्यूटर पे कनेक्ट करना पड़ेगा
Sir, can I say something?
हम्म
Sir, yesterday, after getting that link, I installed that software. I don't know whether it is fully installed or not, but can I say how what I did?
yeah you can tell
Uh, sir, in the Google folder which you have opened right now, where there is a .exe file,
Yeah,
we have to download that file. It takes some time, and after downloadation, we have to run that file. At that time, it
yeah.
need
Just
full internet connection.
yes, it needs
Uh,
full internet connection because
uh,
this is a very small file, but it will connect to that server and it will download the
uh,
other things on your computer.
mm. And after that, system will reboot, and after reboot also, it will again ask to run the same installation, and then
No,
I
no,
think
no,
it will
no,
get
no, no.
fully installed.
It should not. It should not ask you to run any installation. So once you run it, you will get a software like this.
It asked
Okay.
me to how what default it want, proteomics or genomics and what.
No, that's what we will discuss tomorrow. Once you
Okay.
come to this window, that's it. That's what I want. Then I'll show you everything: how to set up the software, how to import the raw files, and how to detect those masses, and then how to identify the molecule and do the quantification.
It
Okay,
has
so
shown
that's
me
what we will
some
we will
different
learn tomorrow.
interface.
Yes.
Excuse me,
Okay.
sir.
So only
Excuse me,
once you
sir. I
inst
have downloaded
yeah,
this
yeah,
app
yeah.
and one window is open and here click proteonics molecular mix. Which option will click?
Just select molecule because we are dealing with molecules now, not
Okay,
proteins.
sir.
So just select
Okay.
molecule.
Thank you, sir.
The interface was different. Okay, sir. Okay.
Okay, so once you have this interface, then from tomorrow we'll start from the scratch and then we'll learn it.
Okay, sir.
Okay.
Okay, thank you, sir.
Yeah.
Now
Thank you
see
very
you
much.
tomorrow.
Thank you very much for your time.
Excuse me, sir. Excuse me, sir.
Mhm.
I have few questions regarding this topic, if you don't mind.
Mm. Yeah, yeah.
Uh, Sir, you have mentioned about the sample size of plant samples.
Mm.
That should be between 7 to 12 samples. So if
Yeah.
I take the each sample in replicates of 3, so there will be any problem
No, no, no.
related?
7 to 12 sample means, say, for example, if you
Hmm.
have one group of wild type or control condition. So in
Okay.
that control condition, you should have at least 7 to 10 biological replicates.
Okay.
Okay, so
क्योंकि
if you are
हाँ
dealing with plants, then maybe 7 to 10 plants. If you are doing an experiment with leaves, then at least try to collect 7 to 10 different leaves.
ओके ओके क्योंकि
Okay.
हाँ सर, क्योंकि सैंपल जब हम लोग रन करते हैं तो
Hmm.
7 टू 12 साइकिल्स अगर ईच सैंपल्स के लिए करेंगे तो वो मतलब बहुत ज़्यादा हो जाता है और उस हिसाब
वो
से
ज्यादा
तो
होता है,
हाँ
लेकिन
हाँ
प्रॉब्लम ये
हुँ,
होता है ना कि
हुँ,
अ, अगर हमारे सैंपल प्रिपरेशन करते समय अगर
हुँ,
हमने सिर्फ 3 रेप्लिकेट लिए,
हुँ,
इनमें से एक अगर आउटलायर निकल गया,
ओके,
उसकी वो ठीक से अगर प्रोसेस नहीं हुआ, तुम्हारे
हुँ,
पास सिर्फ 2 ही रेप्लिकेट आएगा। तो
यस
तुम्हारा जो
सर,
स्टैटिस्टिकल पावर ही अगर तुम्हारा कम रहेगा, so
हुँ,
that's how you can't determine whether the change you are seeing, whether it's really significantly changed or not. To
हुँ,
determine that thing, you need to have more statistical power. I know it sometimes means more money, more effort, but that saves you all the time if you have if you start with a little bit more.
ओके एंड सर जैसे हमारे पास सैंपल प्रिपरेशन जो है अगर डायरेक्ट हार्वेस्टिंग के बाद अगर हम लोग प्रिपेयर करें रादर देन फिक्सिंग इट इन लिक्विड नाइट्रोजन सो
Hmm.
इट वुड बी एनी
Yeah, कर
प्रॉब्लम और
सकते हैं कर no
हाँ
no no कर सकते हैं,
हुँ,
but the thing is that, say
हुँ,
for example, if you are a botanist, if you are working with
यस
plants,
सर यस
okay,
सर
and now you have an experiment where you have maybe 20 plants in one group and 20 plants in another group.
ओके
Do you think that you can process all the
नो
40 plants in 1 hour?
नो सर
Not possible. So
ओके
that's why we need to take all of them, harvest everything, and
हुँ,
put it into -80 so that their metabolism will be freeze, and then you can take 4 or 5 samples every day and then grind them and whatever you want to do, you do it every day. That gives you enough time to do it properly. If you try to squeeze everything in 1 hour or 2 hours time limit, you try to rush through things and you screw up everything.
ओके
You know what I mean?
यस
That is the reason why you need to do systematically, and that's why it's beneficial to save the sample and then process it later on.
Okay. And can we use the nitrogen gas for drying purpose, sir? Because we have
Yeah,
we
you
don't
you
have
can you
speed
can
vac in our department. Mm.
if you don't have speed vac,
Yes, sir.
so you can use nitrogen. So what you can do is that you can keep your sample on heating block, like maybe 35-40 degrees, and you can continuously sparge at a very low flow the nitrogen in the tube or in your container. Okay, so
Okay.
what will happen is that that heat will remove all the water content,
Mm.
but because while it's removing the water content, we don't want our uh, material to oxidize. So that's why we are putting on inert gas over on the top of it so that it should not oxidize quickly.
Okay. Uh, and just few more questions, sir. Okay.
Mhm.
Uh, and for sir, data processing, you have said we have to convert in the open format, and
Yeah,
there is
it
one
depends.
process.
Yeah. It depends. So say for example, if you have uh, a machine from a particular vendor or particular company,
Mm.
and a particular those companies will provide your software, but they will charge you a very huge license fee. So
Okay.
if you can pay their license fee, you can use their software, or you can use a free software which is available on the
Mm.
internet. But that software sometimes will not work with the company's file format. So what we need to do is that we need to convert that file format to a format which that software will recognize.
Okay.
So I'll show you that as well.
Mm.
There is a converter which we can use, and it can convert all the files different from different companies into a format which the open source software can recognize.
ओके। और सर अगर सपोज़ डिफरेंट इंस्ट्रूमेंट्स हम लोग यूज़ कर रहे हैं और उसको सेम ओपन फॉर्मेट में हम लोग कन्वर्ट कर रहे हैं, तो विल बी देयर एनी एनी डिफरेंस और नॉट?
No. It won't be any difference.
ओके।
That's
और
the
सर
whole
हाँ,
point. That's
ओके,
the whole point because uh, these days what happen is that whenever we submit any study uh, to any journal, so what we need to do is that we need to submit all our raw data and the analysis as well to them, so that
यस,
the people who are reviewing the paper or the study, they can look into the data, they can do whatever they want at their end to make sure that you are not faking it, you are not plagiarizing it.
यस,
ओके, तो वहां
यस
पर
सर,
किसी को उल्लू बना नहीं सकते। सब तुमको जो कुछ भी ओरिजिनल चीज तुम्हारी है, यू हैव टू
हुँ,
प्रोवाइड टू द पीपल।
यस। And when we have raw data, sir, then after that we have to do a lot of processing of that raw data.
या या
Just as you say, we have to convert in open format and do
हम्म,
deconvolution and do normalization. So, is
हम्म।
there any single platform where we can perform all these steps, or we have to use different
देर इज
platform?
नो सिंगल प्लेटफॉर्म, देर इज नो
Okay,
सिंगल प्लेटफॉर्म
okay,
Chaitanya. We have to use different tools, different methods, uh, and it takes little bit of time, but I think everyone can learn it.
okay. और सर अगर हम लोग नॉन टारगेटेड मेटाबोलिमिक्स की स्टडी कर रहे हैं, तो
Hmm.
पहले तो सिर्फ जो मास स्पेक्ट्रोमेट्री रहता था, सिंगल मास स्पेक्ट्रोमीटर, बट अभी टेंडम मास स्पेक्ट्रोमेट्री आ चुका है, उसमें भी बहुत सारे वेरिएशन हमको दिखते हैं,
So
टीओएफ या
tandem
फिर
mass
आपने जैसे और
okay.
हाँ,
So tandem mass
ऑर्बिट रैप, हाँ,
spectrometry means yeah.
हाँ,
So tandem mass spectrum means
हुँ,
uh, you fragment those molecules. That's the
हुँ,
tandem mass spectrometry.
हुँ,
Okay.
सो हाँ, सो
So
व्हिच
actually
विल बी
मेरा plan
हाँ,
यही था कि
हाँ,
actually that was my plan कि give you a the workshop. In this workshop I was planning to do untargeted metabolomics, but then I realized maybe a lot of people will not like it, or it will be too complex. That's why I thought maybe do a simpler one which is untargeted.
okay. So, of your opinion, which will be better for untargeted studies? There are various
Both
spec
see
can mm,
both the studies have their
mm,
own pros and cons. In
mm,
untargeted study, you
mm,
don't have to know anything. You just run this experiment, and the
mm,
experiment will tell you what your hypothesis could be, what is the differences, and what you can learn from it. Whereas in targeted mode, we have to know what we want to study.
Yes.
Okay.
Yes, sir.
Just to say, for example, when you run any gene expression study like microarray or RNA-seq, so we really don't know what exactly we will expect. But once we finish that, and then we found that okay, certain pathway are changed, and this pathway is important. So to verify that, what we do, then we select few gene target, then design a primer, and then we do a qPCR on it to verify what we are seeing in the RNA-seq or microarray experiment is true or not.
Okay. Okay, thank you so much, sir.
Okay, no problem.
Okay, thank you, Nishi sir. Now,
Okay.
now I can say good night to you. See
Okay,
you
okay,
tomorrow,
okay.
sir.
Yeah. Good night, good night.
Okay, on behalf of all the
Yeah,
participants, I
see,
would like
yeah,
to thank you for elaborate
yeah,
discussion. Thank you, sir.
yeah. We will meet tomorrow. Okay.
Yes,
Yeah.
sir, definitely.
Okay, sir.
Thank you.
Thank you. Good night, sir.
Good night.
Okay, thank you, sir.
Thank you very much.
Thank you,
Have a
sir.
nice day.
Thank you, sir.
Yeah.
Thank you, all of you. Our next session will start from 1:30.
Okay.
Thank you, sir. Thank you. I am ending the meeting, friends. See you at 1:30.
Thank you, ma'am.
Thank you.